Hello everyone and welcome to Epilepsy Research UK's Research Roundtable on epilepsy treatments. My name is Caoimhe Bennett and I'm the head of research at Epilepsy Research UK. We've got a great session lined up for you all today. So last month, as some of you may be aware, we launched our A Life Interrupted campaign to highlight the interruptive, disruptive impact of epilepsy, the inequalities in research funding, and to call on people affected by epilepsy to shape the future of research. And we have been absolutely bowled over by the responses so far to the Shape Epilepsy Research Network. And we spotted some consistent themes from people who have joined. So when asked how epilepsy interrupts lives, people most frequently reported disruptions to daily life, such as the ability to drive, but also impacts such as career choices. 1/3 of people said that better medications with fewer side effects would be life-changing for them. And over half said that reduced drug side effects, more effective drugs, and new and improved treatment options were their highest research priorities. So we thought it was particularly important to host a research roundtable on epilepsy treatments. But how about all of you watching from home? Do you agree? Maybe you disagree. We want to build the biggest ever community of people living with epilepsy to influence and help shape research. So if you're affected by epilepsy and want to help shape research, then please do consider joining our network. The link for the network will be posted on the Q&A box and is on the screen now. Or sorry, that will be in the chat box. But for now, we have a brilliant webinar to get on with. So we'll be hearing from Professor John Paul Leach from the University of Glasgow, who will discuss the history of anti-seizure medication development through both accident and design. We'll be hearing from Professor Richard Baines from the University of Manchester, who will discuss his current Epilepsy Research UK-funded work and the present-day drug discovery pathway. And finally, we'll hear from Dr. Rob Wykes at UCL and the University of Manchester, who will discuss his research using a material commonly used in the electronics industry and how this may lead to exciting future treatments. But first and foremost, we'll be hearing from Nicole, who will be sharing her personal experience of epilepsy. Following the talks, we'll have some Q&As, so we hope that you can stick around for that. But for now, I will hand over to Nicole. So thank you for joining us. And here we go.
So I'm Nicole, I'm a mum of two. I've got a boy who's 4 and a girl who's 1. And our journey with epilepsy started in July of this year. So our 4-year-old boy was in the bath and he was just playing with his sister, and then suddenly the colour drained from his face. He went rigid and he started tipping. We quickly got him out of the bath and put him in the recovery position. He was unresponsive for 3.5 minutes, and it felt like a lifetime. I'm sure any of you who've experienced seizures and seizures of your children, I was scared, worried. It was awful seeing that. We went to hospital and he was diagnosed with first seizure, and we went away with some seizure first aid, and we were told to just watch and wait. 2 weeks later, he had 2 seizures in very quick succession. One was on the toilet, one was in bed. Again, that colour draining of the face, is such a vivid, picture for me. The only other time that I've seen that is when I saw my granddad, who was dead, and that's the colour that I saw. So, just awful experiences there. Through this time, we're just praying, for God's peace and just protection. We were unsure, what was going on. We went into hospital and we stayed there for a few nights and had lots and lots of tests, and then had a 48-hour EEG, and we were told that there was abnormal activity in the left side of his brain, and he was actually diagnosed then in August with focal epilepsy. Now, like I said, I didn't know anything about epilepsy. I thought maybe it's one thing, but actually so many different things. You know, I've learned so much, you know, through this time, and you know how common it is for people to have a form of epilepsy, but yet you know how little you know funding is put into the research. We, you know, our life is interrupted daily. You know, as a mum, we worry about our kids anyway, but actually I've just been on heightened alert ever since. You know, watching him sleep, you know, the worry of sudden unexplained death and epilepsy. His seizures aren't controlled at the moment, and yeah, so all of those worries, just constantly watching him on the toilet, different things like that, and trying to look after my 1-year-old at the same time. So you know, I'm someone who tries to go for natural support, but because of the complex nature of epilepsy, I definitely did not feel qualified to say no to medication. So we started looking into medication. There was complexity in the decision because my cousin's son had a severe reaction to one of the drugs that was suggested. So we ended up starting on Keppra. Now, I read the side effects because I want to be prepared and just, you know, look out for the things to look out for in my son. And I cried reading those side effects because I saw that 1 in 10, depression, anxiety, rage. I did not want to lose my son to that rage, to, you know, for his mental health to be affected. He's only 4, and, you know, he just started school. But anyway, we went on that. Unfortunately, we have seen an increase in rage, and we're actually starting weaning him off Keppra because you know, it's very intense. The side effects are very intense, and we want to try and find that you know, that way to try and control the seizures, but also not impact him as an individual as much. So yeah, we've seen his personality, you know, being lost, and we really want to see him back. Last week we started on a new medication, and I feel like looking at the medication and looking at the different side effects, I feel like is you know, weighing up what's the lesser of two evils, or what's the least worst side effect, and you know, because you don't want any side effects, but you don't want the seizures. I don't want him to keep having seizures, but I don't want his mental health affected. I don't want this new one is potential to cause heart irregularities. So as a parent, trying to weigh up that decision and make that impact, you know, that's going to impact him. We don't know if it's going to be a lifelong condition. We don't know whether, you know, he may grow out of it in his teen years. We're just going to have to wait and see. But you know, I'm definitely super excited to hear about the research that Epilepsy Research UK are doing, because actually, you know, the impact of the side effects has already played a massive part in our lives, and I know it affects so many people with epilepsy. You know, as a parent, you want to take away any hurt, but you can't, and so, you know, we just need to go through this new medication. He's actually been seizure free for 10 days now, which is exciting, and start the weaning process of the previous drug to hopefully get our son, our sweet little boy, you know, back to more of himself. So yeah, that's our journey so far.
Thank you so much, Nicole. Thank you so much for sharing you and your family story with us. I know I definitely find that quite an emotional watch, and we will be coming back to you with some questions later on. But for now, just a huge thank you for sharing your experiences. That's hugely appreciated. So thank you, Nicole. And next up, I would like to introduce Professor John Paul Leach, who is Professor of Clinical Neurology and Honorary Consultant and the Head of Undergraduate Medicine at the University of Glasgow. So over to you, John Paul.
Eva, thanks so much for the introduction, and thanks so much to ER UK for the opportunity to do this. I think the possibility of opening discussions with patients and other professionals is so important, and there's always something so moving about hearing accounts. There's something about being a clinician. When you hear accounts within clinic, you can put that in the professional box. When you hear accounts out with clinics, I always find it so moving, and thanks very much for that, Nicole. I think it speaks so much to the needs that we still have around epilepsy care in the UK and in fact across the world. I just want to have a wee quick word about treatment of epilepsy and the drugs that we tend to use and the other methods of treatment that we have, and just a wee talk through these and talk through what we know, and possibly more importantly and more excitingly what we don't know, and the known unknowns are what might open the door to proper treatment for epilepsy in future. There's going to be more to come from this from our speakers and to come, but I just want to say this, that actually we know that in the brain there will be an excess of excitation and a reduction of inhibition, but for most patients we don't know what the molecular mechanisms are that will cause this. Although we know that in more than 2/3 of patients the cause of epilepsy is unknown, there are even more mysteries. If you have two patients with similar brain injuries due to head injury, only one will develop epilepsy and one won't. What are the molecular mechanisms that underlie this? And this lack of knowledge around it is something that's going to bear much fruit in our future investigation. I think you're going to hear more about that in the talks later on. As a result of the fact that we don't really know the molecular mechanisms of what underpins many patients with epilepsy, we find that actually the treatments tend to have been stumbled upon. And the history of anti-epileptic drug treatment often involves drugs that are used for something else, usually for sedative purposes. And in their use, they sometimes find that seizure counts are reduced in patients who are exposed to them. And that particularly underpins the use of the bromides and the barbiturates. There was beginning to be some design around treatments, and they'd looked specifically, once they knew how action potentials are formed in the brain, they began to develop drugs which affected and reduced the flow of sodium currents, the so-called sodium channel blockers. Sodium valproate was another drug that was found completely by accident in some experiments, where it was initially used as a solute for testing materials, and in testing the materials they found out that the solute itself had very marked anti-epileptic activity, and the early drugs were largely stumbled upon rather than designed. By the time we got to the later drugs, we find that more of them have been sought out. We've found a molecular mechanism. We've tried to design a molecule that will impact on that molecular mechanism. And we're beginning to see a wee bit more design, a wee bit more intent in some of the molecules that we have. And these are not perfect, but they're beginning to offer us more and more opportunities. And we see here a graph that is very imposing, very intimidating for some clinicians who are not specialists. And of course, for some, they see this as disheartening. We've gone from a handful of medications in the 1980s to well over 25. And of course, there are further drugs with different mechanisms of action on the horizon for use in the broad clinical setting. So this is something that, as an epilepsy consultant, I think is very heartening. But of course, there is no point in just building up a large store of drugs, all with different mechanisms of action, if we can't use them sensibly. So the early care in the 1980s, when I was a medical student, really involved reviewing patients, looking at the seizure diary and using a syringe, sometimes to take blood samples to measure blood levels. And there was reason for that with the older anti-epileptic drugs, but really there was not much else to offer. We had a small number of drugs, and these drugs tended to interact quite markedly and tended to cause quite marked effects on liver function. But as we've moved on, and this is thanks to much of the basic science that was carried out in the 1980s and beyond, the 1990s, as you might already know, was deemed to be by the WHO as being the decade of the brain, and this led to a massive increase in funding to look at mechanisms of disease in neurochemistry and epilepsy and other neurological diseases, and also looking at mechanisms of actions of drugs that may in some way impact on it. And this massive investment began to pay off and actually underlay a lot of the acceleration in treatment availability that you saw in that previous graph. Where are we with the new drugs? What are they good for? What are they giving us that they weren't giving us before? Well, we've got a wider range of actions that we can pinpoint, which might be useful if more information becomes available. They're certainly much more stable in the bloodstream and tend to interact a lot less severely than in the older medications. They're more stable in the bloodstream. They have a longer half-life, so there's not so much variability in the levels in the blood. There are fewer, it would appear, long-term side effects. They have less long-term action on bone health and on liver function, so therefore we expect the metabolic effects of decades of treatment will be much less with our older ancestor drugs. It would appear that for some we get more information available that they're having less effect on the developing fetus, therefore making them safer in pregnancy, and the more information we get about that, the better. And of course, for some patients who have very frequent seizures or prolonged seizures, we need a way of administering drugs that is easy in the community and can treat prolonged and severe seizures. And we're beginning to get more options available for that. But what hasn't changed about the new drugs? And when I look at what patients want from their medication, one of the things that you saw from what Caoimhe said is they want more effective medication. Now, this is a landmark study, Brody and Kwan, back in the early 2000s. And this showed that around 2/3, 70% of patients or so will be fully controlled on the first or second drug that we try. Now, that in some ways is good news. We can tell patients that the most likely thing is that they will be seizure-free. But even with the advent of the newer drugs, we haven't seen this yellow triangle here, we haven't seen this shrink too much. That although we've got a greater number of drugs, we still see just as many patients who are continuing to have seizures. So although our drugs, I think, are better tolerated and have lots of good aspects to their use, they're still not managing to render the vast majority of our patients seizure-free. So there's what I would term in school terms, room for improvement here. How do we in the clinic tend to choose anti-epileptic drugs? And I was very struck by Nicole's story about a discussion with patients and with families around which drug we'll choose. This is absolutely vital. But what we're left with is making a series of choices, picking on drugs depending on the type of epilepsy, whether or not the patient is due to or possibly going to become pregnant in coming years, whether there are any pre-existing symptoms that might be worsened by anti-epileptic drugs, and of course sometimes side benefits. Some anti-epileptic treatments can have very positive effects on mood or may help with weight loss and may help with other aspects of healthcare. And of course the ease of administration, a big factor in your treatments, is something that we're going to look at here. So we will take lots of these factors into account, hopefully in discussing with the patient and their family to make a decent choice. Of course, sometimes patients turn around and say, well, you've given me 3 or 4 options here. Is this just trial and error? And unfortunately, there is an element of that. And we've heard this from Nicole, where the first drug that was tried is having side effects, and moving on to a second drug hopefully appears to have better effects and less side effects. And of course, sometimes I like to think it's not just guesswork, there's a bit of education, and I usually like to feel on the whole that the education outweighs the guesswork. And I try to convince patients that as we make a rational choice of treatment along with them to help plan treatment choices, that will make things better. So how do we want to choose? What are the options in the future? And I think this all comes down to more and more information about the molecular mechanisms underlying each individual's epilepsy. There's no point knowing how cohorts behave. I want to know how the patient across from me is behaving in a neurochemical way, so that we can target that best with drugs. Now I want to know which mechanisms underlying it that will make full control of seizures more likely with anti-epileptic medications. I want to know which mechanisms of action are innate in them that are already helping their own brain's defences. And I want to know how each individual drug is going to respond with that person's brain to either cause side effects or just selectively affect seizure incidence. So there are lots of questions we need to know, and the research is going to unfold in the coming years and the coming decades, hopefully meaning that we'll make treatment choices based on an actual individual's need and an individual's neurochemistry, rather than making an educated guess. So I'm delighted to start this conversation today. I'm delighted to answer any questions that you may have. So feel free to pose these just now. Thanks, Caoimhe.
Thanks so much, John Paul. That was a brilliant whistle-stop tour of epilepsy medications. So thank you so much. And we've got a few questions to ask you. You've got to rush on after the webinar. So, we're just going to, we're seeing Richard's slide at the minute, but if he could just take that down, please. Thank you, Richard, sorry. But first and foremost, we thought we'd ask you the flip side of some of the questions that we're asking people as part of the SHAPE network. So in your experience, how do you think epilepsy interrupts people's lives?
I think Nicole's very well described the anxiety around this that affects patients' lives and their families' lives. One thing that strikes me is the patients' and families' ability to heroically deal with the unpredictability of epilepsy. We take for granted that when we leave for work in the morning or we go shopping, that we will return in the same state whenever we want. And patients with epilepsy and families of patients with epilepsy have the unpredictability in the background that makes seizure control so important.
Oh, thank you. And secondly, is there one thing that you think would be life-changing for people with epilepsy?
I think your survey encapsulates this perfectly. And although effectiveness against seizures is important, I think as clinicians, we often forget that actually freedom from side effects is important. These drugs have lots of different effects on brain chemistry and brain action, and the less drugs that we have and the more specific the actions of drugs, the less the side effects will be. And this is what we strive for, not just in finding new treatments, but also in finding ways to use older treatments as well and helping to target those to the patients who will do best and suffer fewest side effects.
Brilliant, thank you. And we've actually had a question submitted from a viewer who is also called Nicole, but I don't think it's our same Nicole. And they were wondering, what about the ketogenic diet as a treatment option?
The ketogenic diet is recognised as a treatment for epilepsy. I have to say there are some specific genetic conditions, particularly GLUT1 transporter deficiency, where the ketogenic diet is highly effective. Some people think that it's similar to Atkins diet. It's actually quite a bit more difficult and quite a bit more demanding than the Atkins diet. And of course, restricting carbohydrates so severely is a difficult thing to sustain in the long term. But for some patients, it can help improve epilepsy. But I would say that it tends not to be a treatment that we would roll out widely in the epilepsy clinic. I would reserve it for patients usually where other different drugs have failed, and I would reserve it usually most specifically for patients with definite genetic abnormalities causing their epilepsy.
Thank you. And final quick question, what are your hopes for the future of research into epilepsy?
I think you're going to hear about some fantastic research into epilepsy. And whenever I go to the ILE and these treatment conferences, I see lots of different avenues towards it. But what I would really love is the ability to speak to a patient and say, you have a problem with X mechanism and Y drug is going to fix it. I would like more specificity. I would like particularly to be able to predict who's going to be seizure-free and when, and who's going to receive side effects and who won't. And that way we can take a lot of the guesswork out of it and leave us with just the education and planning of treatment.
Brilliant. Well, I know what'll be on your Christmas list then. So thank you so much, John Paul, for joining us. It's really been brilliant to hear from you. So thank you.
Thank you, Caoimhe. Thanks. And goodbye, everyone. Thank you so much.
Take care. Now we're handing over to Professor Richard Baines, who is Professor of Neuroscience at the Division of Neuroscience and Experimental Psychology at the University of Manchester. And for those of you who have been on our website, Professor Baines's blog was featured yesterday and was a fascinating whistle-stop tour through the drug discovery pathway. So over to you, Richard, and thank you for joining us today.
Well, thank you, Caoimhe, and thank you to Epilepsy Research UK for giving me the opportunity to join you today. And thanks to Professor Leach for giving me a very good introduction into what I wanted to talk about today. Professor Leach has talked about the almost 30 drugs which are available and how, as with all drugs, they have benefits, but they have their downsides, and the downsides, Nicole has pointed out very well, in that they have side effects. So the research that my research group looks into is really trying to help the brain to help itself. And I'm going to use a pointer as a typical academic, but this is the human brain here. Inside that human brain, there are 80 billion nerve cells. And all of those nerve cells talk to one another, they excite each other, they inhibit each other. And for the most part, the brain does an enormous job to maintain a balance between excitation and inhibition, and that balance allows the brain to function. But in, let me get rid of that. But in certain diseases, and epilepsy is one such disease, that balance becomes slightly skewed towards excitation. So the brain is easier to excite, and that's the basis of seizures. And in fact, the drugs that Professor Leach talked about act through one of two main mechanisms. They either act to repress or reduce excitation, or they act to increase inhibition in the hope that we can restore the balance back to the level playing field that's characteristic of the brain. Now, my research over the last 20 years has studied how the brain maintains this balance and study the endogenous mechanisms, the inbuilt mechanism that the brain uses to achieve this. And this is called neuronal homeostasis. And homeostasis can be applied to many different biological mechanisms. It means to return to a set point. So we can talk about neuronal homeostasis, we can talk about glucose homeostasis in the blood, temperature homeostasis of the body. It's just a term to mean a balance. And it turns out that every neuron, every nerve cell in this brain, and this is my nerve cell here in black, it fires action potentials, the electrical signals that the nerve cells use to talk to one another, and each vertical line here is supposedly an action potential, an electrical signal. If those nerve cells become too excited for too long, neuronal homeostasis works to make adjustments to those particular nerve cells to bring it back to what we call a set point. And that set point is somehow encoded or set within every nerve cell during early development. So all of the nerve cells in this brain have some knowledge of where they should be in regards to the excitation inhibition balance, and the brain is constantly checking, constantly making adjustments to try to keep that balance as level as it can. But in cases where we carry perhaps a mutation, or we've had an injury or some sort of other external factor, excitation can predominate, and that can inevitably lead to this brain being more prone to seizures. So my research has tried to understand this process from a basic science point of view, but it's also tried to now make capital from that, because if we understand how something works, that's the opportunity to exploit the different components to allow us to intervene with a drug. So we identified a protein, it's called pamilio, it's an odd name, but part of this homeostatic process. It helps these nerve cells to readjust how active they are. And using gene therapy, we showed that manipulating the expression levels of pamilio was a very potent anti-seizure manipulation. However, giving somebody a protein as a drug is not very useful. If you ingest the protein, your gut breaks it down very quickly. It's no different to eating the Sunday roast. It's protein, and your gut will break it down. So what we really need is a drug that tricks the brain into increasing the expression of this protein in the hope that that achieves our clinical benefit. And so over the last 2 or 3 years, with help from Epilepsy Research UK, we have developed now a drug which increases the expression level of this protein. And we've shown that that drug has very potent anticonvulsant, anti-seizure properties. So the advantage of this approach is we're helping an already existing mechanism that the brain uses on a daily basis to try to maintain stability of its function. And so if we can boost this activity, we may be able to guard against seizures, reduce the frequency of seizures, regardless of the underlying cause. And as Professor Leach pointed out, there's many different causes of seizures in human individuals. So I'm now at the start of what we might call the road of drug discovery. And I found this image. It's a long road. We can't see the end of it. And I know many people say and are frustrated by how long it takes to develop a drug. And so I thought I'd just share with you my experience so far, how far we've got down this road, and to give you an idea of the sort of hurdles, or maybe potholes is a more apt description, that face us before we get to the end of the road with a drug that actually finds its way into the clinic. The first step is to really understand what we call the mode of action. This is, how does the drug work? Well, we know the drug increases the protein called pomelio, and that in turn helps this homeostasis to work, and that keeps order in the brain. But we don't know where in the mechanism of homeostasis our drug works, what particular step of homeostasis. And one of the problems here is, well, we don't fully understand neuronal homeostasis. It's still lots of unanswered questions. So this is a basic science question. We need to understand more about homeostasis so we can understand where our drug works. And indeed, that might identify even better drug targets along the way. The second step is to find a drug and to be sure it has suitable drug properties. And again, Professor Leach has indicated some of those. So our drug must be stable to be ingested. It must survive the gut. It must then get out of the gut and into the blood at enough levels so that it can then get into the brain at equally high levels to be effective. How long will it be before the body metabolizes the drug? Hopefully the longer the better, and therefore you can take less drug less frequently. So we are working with medicinal chemists to be sure that our compound also has those suitable drug-like properties, so it actually becomes usable once we finally get to the clinic. And again, Professor Leach talked about the 30% of individuals who are living with epilepsy for which all of the present-day drugs really offer no benefit. And we really want to target that particular population in particular. They have the most need at the moment, but we want a drug which is beneficial across the range, but certainly for the drug-resistant epilepsies. And so we can provide clinical benefit for the 30% of people who struggle to control their seizure frequency or severity. And then, of course, it has to be safe to use. Side effects, those side effects have to be tolerable. They can't be worse than the benefit of the drug, and particularly as these drugs need to be taken for quite a long period of time, if not a lifetime, then side effects are very, very important to ensure safe usage. And this is, I think, where targeting a mechanism that already occurs in the brain is beneficial, because our investigations have already shown lots of checks and balances in that mechanism to make sure it never gets too active or too underactive. So it's got its own built-in safeguards already. What we're trying to do is just to boost it a little bit and not too much. So we're working on all of these aspects, and I would guess that this would take about the next 5 years before we've really ticked all of these boxes. And then once we've got a tick against these boxes, then hopefully we're in a position for clinical trials where we start to move into the clinic and testing our compounds in human patients. And again, that might take 5 years. So all in all, it would take probably another 10 years before we really know whether our drug will make it to the clinic for the benefit of those living with epilepsy. And at that point, I will finish.
Thank you so much, Richard. That was a really fascinating whistle-stop tour. I keep saying that in this webinar, but it's true, of the drug discovery pathway and all the hurdles that you as a researcher have to overcome. So thank you so much, and we'll be back to you with some questions at the end. And so for now, I'd like to introduce Rob Wykes, who is a senior research fellow at University College London and senior lecturer at the University of Manchester, so he's very busy. And Dr. Wykes was an Epilepsy Research UK Emerging Leader Fellow from 2015 to 2018. So we're delighted to have him here to show us what he's up to now. So thank you, Rob.
Okay. So, well, of course, thank you very much for inviting me to the ER UK Roundtable research panel. And I'm going to discuss some of the areas my labs are taking to develop new technologies and treatments for people with epilepsy, particularly those who do not adequately respond to anti-epileptic drugs. But this is really the future. We've heard about the past and the current, and we've heard about the near future. Now I'm talking about a little bit further ahead. So we've heard from the other two speakers the challenges of anti-epileptic drug therapy. The brain is complex and it presents many distinct challenges for drug delivery and effective therapy. With epilepsy, although many people will achieve control of seizures using anti-epileptic drugs, there is an absence of any curative pharmacological treatments, meaning that people will continue to have to take medications for long periods of time or forever. What's more, some people who live with epilepsy who do obtain seizure freedom will also experience a range of comorbidities or side effects, and we heard that earlier today. These can include a variety of things, including sedation, nausea, and headache. And of course, most importantly, there's a population of patients who do not respond to anti-epileptic drugs at all. So there are over 40 different types of seizure, and seizures are different for every individual. And although there are many drugs and therapies available, about 30% of people live with uncontrolled seizures and do not respond to medication. This population is termed drug refractory or drug resistant, and it can affect up to as many as 180,000 people in the UK alone. There are alternatives, treatment options that are not pharmacologically based, and we heard about the ketogenic diet earlier, but there's also brain surgery and brain stimulation. And there are other experimental treatments which we hope are on the verge of clinical translation, and these involve gene therapies. So very briefly, what is gene therapy? Well, it's an exciting area of research which holds promise for several complex neurological disorders, including Parkinson's disease, Alzheimer's disease, and of course, epilepsy. Gene therapy has several conceptual advantages. It's based on a rational design of molecular tools, and it can achieve a regional modification of that area of the brain's excitability. Gene therapy usually involves using specialized modified vectors that can transfer genetic information into cells. As many drug-resistant seizures will have a focal onset, i.e. that means the seizures start in the same restricted area of the brain. Gene therapy can offer the prospect of a localized treatment where you apply that gene therapy just to that area of the brain. Therefore, you might avoid some of the side effects that you would experience from global delivery of anti-epileptic drugs. For the last 11 years, I've been working in gene therapy strategies with colleagues at the UCL Institute of Neurology. And in the department, several different approaches have been developed. Most advanced is a strategy which involves overexpression of a particular protein. It's a potassium channel, which is called Kv1.1. And if we overexpress this in the neurons of the seizure focus, our preliminary preclinical work shows that this can dramatically reduce seizure frequency. The graph there you're showing in blue is after treatment compared to the graph in red, which is without the treatment. And the study leaders of this particular gene therapy, who are Professors George, Walker, and Coleman, are preparing for a human study of this therapy at the National Hospital for Neurology and Neurosurgery. But going forward, are there other gene therapy approaches that could be used for all different types of epilepsy, a more personalized approach, for example, to correct specific genetic epilepsies? And here what we're really talking about is the possibility of gene editing. And indeed, the Nobel Prize this year was awarded to researchers who discovered a specific gene editing pathway. I would like to state really at this moment that this technology has been shown to work very well in individual cells in a Petri dish, but there are considerable challenges to overcome before these approaches will be able to repair genetic mutations in living people. And I'm happy to discuss that later. For many treatments, from surgical resection to brain stimulation to localized gene therapy approaches, it is absolutely crucial to be able to detect where in the brain the seizures are starting, the seizure onset zone. This is currently done by a mixture of techniques. One can obtain clues from the semiology, the signs or symptoms that occur during a seizure, from identifying brain malformations or lesions using imaging approaches, but perhaps most importantly, it's used by performing EEG recordings of brain activity. So there are several brain activity biomarkers that can be used to identify seizure onset zones. However, the metal-based used for this do have some intrinsic limitations, and they're not well suited to record some biomarkers which we know exist, for example, a DC shift. Therefore, could alternative materials be better? And for the last couple of years, I've been working on the development and characterization of different types of brain probes that are based on graphene. And we think these are going to benefit both epilepsy research and potentially treatment going forward. So I'm going to quickly play, it's just a brief 1-minute video, which illustrates some of the advantages graphene has. Clearly, not all of these are specific for the probes that we're developing for epilepsy, but it'll give you a brief idea. So there we go. And on this slide here, you can see some of the schematics and photos of different types of graphene-based microtransistors and electrodes that are currently being evaluated for their use in epilepsy research in my labs. For example, a validated biomarker that is seldom used due to current technology limitations is the presence of altered electrical brain activity just before a seizure, which is called a DC shift. And we have determined that our graphene transistors are capable of recording this altered brain activity, as well as other electrical biomarkers. The ability to record DC shifts, coupled with improved MR imaging compatibility that graphene electrodes allow, may mean that we're able to develop clinically applicable graphene probes that will more accurately identify where seizures are coming from. And this has the potential, therefore, to improve the accuracy of surgical resection and for localized gene therapy strategies, leading to improved success rates. In the final 2 slides, I'd like to also mention neurostimulation or brain stimulation. This is an alternative treatment option for patients with various neurological disorders, including epilepsy, that don't respond to medication. And various methods of brain stimulation approaches can be used experimentally in the clinic with promising results. For both brain stimulation and recording, though, electrodes are required to be small, sometimes even smaller than the diameter of a single human hair. And they cannot provide enough charge to produce the response without irreversibly damaging the electrode material. And this results in probe failure over time. In contrast, graphene microelectrodes can be made in such a way that they have an increased electrochemical surface area. And that means they can provide enough charge without damaging the electrode, and they can reliably stimulate for much longer periods of time. Here, if you see the blue line, you can see how stable graphene-based stimulating electrodes are compared to alternatives. For example, graphene probes can stimulate over 110 million pulses and inject over 200 times more charge than platinum-based electrodes. Finally, I'd like to talk about spreading depolarizations and their potential role in SUDEP. So spread and depolarizations are associated with several neurological diseases, including migraine, brain cancers like glioblastoma, traumatic brain injury, stroke, and of course epilepsy. And it's estimated that about half of all epilepsy-related deaths are caused by sudden unexplained death in epilepsy, which is called SUDEP. However, little is known about the underlying mechanisms that can result in SUDEP. In recent years, a proposed mechanism, not all the mechanisms, but a proposed mechanism, is that SUDEP is caused as a result of spread and depolarization. This is a pattern of activity that occur in the brain after a seizure. Essentially, this means that the brain will experience a sudden wave of hyperactivity followed by a wave of suppression of brain activity. Worryingly, this inhibition can affect areas of the brain that are required for breathing. Traditional metal-based electrodes, shown in red, are unable to detect these events during recordings of brain activity, and so they've been poorly studied in the context of epilepsy. We are now using graphene transistors, where the traces shown in black show this activity, to study spread and depressions after seizures, gaining information on how they occur and how they might be treated. So I hope this short talk gives you an idea about some of the epilepsy research directions going on in my lab. I'd like to thank all my collaborators and of course the people who are funding the research employed in Epilepsy Research UK. Thank you very much.
Thank you so much, Rob. That was a brilliant talk explaining some really complex research that you have going on. And we'd love to invite Richard and Nicole to join us again for some quick questions. We've got just over 10 minutes of questions. And we've had some great comments in the chat section and also on Facebook of people saying that, you know, this research is really interesting, but also hopeful, which I think is really nice. And I'd like to begin the questions by asking you, Nicole. You touched on this as part of your talk, but I'd like to ask you some of our SHAPE network questions. How has epilepsy interrupted your life and your family's life?
It's interrupted our lives in lots of ways. So when I'm with my son, I'm just constantly watching him because we're not quite sure of the triggers right now. So he needs to be, you know, so that interrupts. And when I'm not with him, I'm constantly looking at my phone to see if I get a call from school that he's had a seizure. You know, it disrupts the family dynamic with the side effects as well, has been very disruptive. And for our 1-year-old daughter to watch her brother going through these things.
Of course, I know some of my colleagues were saying they really find your talk earlier really emotional. So thank you again for sharing your experiences. And is there one thing that would be life-changing for you and your family?
Yeah, the finding, so it's really, really interesting to hear about all this research. It's amazing. I've learned so much. And, you know, there's that hope, isn't there? That hope for the future, that the research will find a treatment that you know, helps everyone be seizure free. That would be life-changing. And also without the side effects that a lot of the anti-epileptic drugs have.
Of course. Thank you so much. And this is a question for Richard and Rob. We've had a great question from Sharon on Facebook. So we're currently seeing scientific research happening at an extremely accelerated rate in relation to the COVID-19 pandemic. It seems like every time you switch on the news this week, there's a new breakthrough with a vaccine. But why do you think this isn't happening with epilepsy? And why does scientific research take so long? So perhaps, Richard, if you'd like to answer first.
I think the first thing to say is, you know, COVID is a worldwide emergency. Huge amounts of finance and huge amounts of manpower have been instantly targeted to COVID. So all of the big pharmaceutical companies have stopped their programs and jumped into the search for a better vaccine. Governments have pumped in billions of pounds, and it will continue to do so. But it's also worth noting that actually vaccine technology is reasonably well established. And I don't want to diminish what people have achieved because it's absolutely amazing. But we do know the established technology is just trying to re-engineer it for COVID. And it's technology that we're using every year. We make flu vaccines every year. There's some tweaks and some twists, but it's pretty much basic, tried and trusted. Whereas in epilepsy, the amount of funding that's going into epilepsy is far less. One could argue there's less of a population. There's 1%, I think, of the population in the first world that have to live with epilepsy. But I think epilepsy does get less share of its research than other diseases such as Alzheimer's. And I think Epilepsy Research UK and these sorts of webinars will hopefully go to some way to address that imbalance. But it comes down to money and manpower at the end, because the work is expensive, and the more people you have, the quicker things get done.
Thank you, Richard. And you're quite right. We conducted some research as part of our Shaping the Future of Epilepsy report. And it really shows how the stark inequalities in terms of money to epilepsy research funding. It's been chronically and critically underfunded, I think. So, Rob, same question to you, please.
Yes, I have to agree with you that obviously I think the biggest cause is the lack of funding that epilepsy receives compared to this. But it's also a different type of disease. So with COVID, there's one virus, there's one kind of mechanism really that you're trying to prevent, is the infection of that virus. And so I could understand why many groups would be able to close in on that in a relatively short period of time. And as Professor Baines was saying, that people who work on flu vaccinations are already ahead of the game in that. But with epilepsy, it's really an umbrella term that covers an awful number of underlying causes that can all end up resulting in seizures. And a particular drug or a particular therapy is not going to be the magic bullet for all types of epilepsy. And therefore, again, our kind of research is being split amongst lots of different areas of epilepsy research as well. Because I don't think there is going to be just one thing which will cure everybody of epilepsy.
Of course, that's so true. We always talk about epilepsy as one condition, but you're right, it's that umbrella condition, and there's so much more to it than that. And so, Rob, whilst I've got you here, I'd like to ask what first got you interested in epilepsy research?
Okay, so, well, I'm a scientist. I'm a basic scientist, PhD, but I've always worked in neuroscience. And I have to say, it was about 15 years ago when I was working in America that it was just when we were starting to be able to use viral-based vectors to manipulate neurons in the brain. And I felt that this was going to be a really exciting area, but also potentially in terms of therapeutics, something that could be going down the line very important. And just serendipitously, as that postdoc position was finishing, there was an advert by Professors Coleman, George, and Walker at UCL asking for people with experience with gene therapy approaches to join their lab to start developing epilepsy gene therapy. And so it was a natural progression to actually move into that field. And I'm very glad I did.
Brilliant. We're glad that you did too. And Richard, what got you interested in epilepsy research?
So again, it's a sort of a similar story. So I'm a basic scientist, and I've spent 30 years really studying how nervous systems develop. How do all of those individual neurons come together to form the networks that form the brain? And very much in the early days when neurons are developing, the amount of change that they're undergoing is quite extreme, because they're building themselves, they're making networks, they're talking to one another. And neuronal homeostasis, which I've studied mostly during my career, is so important during those early stages to make sure stability emerges from that group of nerve cells which just come together. They don't have a guidebook how to do it. They have to work out how to communicate. And so once we understood neuronal homeostasis, I started to consider then surely we could use this and exploit it. And then I started to talk to epilepsy consultants, to epilepsy researchers, and I've now not only worked from the bottom up, the basic science, but I now work to study seizures downwards to give me sort of two roads in to the same problem. And I think science really benefits from actually approaching a problem from multiple angles.
Thinking outside the box. That's Brilliant. Thank you so much for answering those questions. We've just got time for one final question, and this is for all three of you, but I'd like to ask Nicole first. What are your hopes for the future of research into epilepsy?
I'm hopeful that more people like Dr. Rob and Professor Richard do more research into those things that will mean that there's less disruptive lives for those with epilepsy, and so they're free to be who they can be and live that full uninterrupted life.
Thank you so much, Nicole. And Rob, what are your hopes for the future of epilepsy research?
Okay, briefly, I see it in two parts. I see one part which I think was touched upon by every speaker today, which was kind of like the personalized aspect of that. There's lots of different mechanisms that can result in seizures. And if we can identify what those mechanisms are, if we can identify a drug which acts specifically on that mechanism without side effects, or we use a gene therapy approach for that specific mechanism, then I think that's going to have a much better outcome. And then the other side, I think, is the technology development in terms of detecting seizures, identifying seizure onset zones, having automated seizure detection programs, wearable headgear, which can let people know in advance if they're likely to have a seizure and therefore, take, precautions. I think both of those strategies are equally going to be effective in managing people's lives with epilepsy.
Of course, and that ties in with one comment on Zoom that we've had from Alistair, talking about whether progress is more promising in either technology or medicines research, which is, it seems like
they're both very useful areas.
Yeah. And so, Richard, finally, what are your hopes for the future of research into epilepsy?
I think my hopes are slightly looking further down the road. I think if we can predict who will go on to develop epilepsy, we will be able to intervene before seizures first start and actually prevent epilepsy and provide a cure. Thereby, people will become drug-free for the rest of their life. And I think the first experiments to show that that may occur have now been published. So I think there's real optimism looking 20 or 30 years down the road.
Brilliant. Well, thank you so much to all of our speakers, to Professor John Paul Leach, who had to dash on, to Professor Richard Baines, and to Dr. Rob Wykes. But most of all, thank you so much to Nicole for sharing your personal story. It was really, really touching, and I hope we've kind of left that on quite a hopeful note. So I'd like to also thank all of our attendees for joining us today, and thank you so much. So we'll hopefully see you at the next Research Roundtable. Thanks and take care.
Thank you very much.
Thank you.